Hello guys, welcome back to Dr. His Academy. Today we'll be discussing an important aspect of metabolism known as urea cycle. Stay tuned. So why do we need to produce urea? Because we ingest a lot of proteins every day, and these proteins can be broken down. So we have our proteins being broken down or digested to form what? Amino acids. Then the amino acids need to be further broken down to form what? Carbon skeleton. So it's this carbon skeleton that is used to what? Generate energy, right? And then it puffs out what? Ammonia. Because amino acids consist of what? A carbon skeleton and an amino group. So this ammonia is very toxic to the body. And then the ammonia needs to be what? Incorporated into a less toxic or a non-toxic form by the liver, known as what? Urea. So we have our proteins entering the GIT, and then this protein will be digested to what? Amino acid, right? And then the amino acid would move into the blood, and then what? The tissues. So it goes to the rest of the tissues, right? The cells of the body. And then we have our amino acid over here. When the amino acid has come to the tissue, so let's say this is your tissue, or this is tissue cell of the body. So this cell would represent most tissues of the body, right? Now, the amino acid is in the cell. What happens? The cell thinks, okay, do I need protein? If I need protein, then I have to use this amino acid to generate what? Protein. So if the body does not need protein at that moment, what happens? The body needs to convert this amino acid into what? A carbon skeleton, which it can use to what? Generate energy, right? Now, this amino acid is special in the sense that it has an amino group. This amino group prevents it from being what? Broken down, all right? So the body has to yank off the amino group in the form of what? Ammonia to generate what? A carbon skeleton from the amino acid. This carbon skeleton can now enter the what? TCA, that's the tricarboxylic acid cycle. You can check the video. I'll put it in the description below. So it enters the what? Tricarboxylic acid cycle to be further broken down into what? Energy, all right, and CO2, okay? So this amino acid will be broken down to form what? A carbon skeleton, known as what? Your alpha keto acid, right? Okay, and then the way it does it is through a reaction called what? Transamination. Transamination. So your cell takes the amino group from amino acid and then dumps it into what? Another carbon skeleton known as what? Alpha ketoglutarate. So we have the what? Alpha ketoglutarate. Okay, and now this reaction is called what? Transamination reaction. That's trans of amino group from one molecule to another. So transamination, transfer of amino group from one molecule to what? Another. Now, the amino group will be obtained from what? Amino acid, right? And then it's being transferred to this boy over here, known as the what? Alpha ketoglutarate. Then the alpha ketoglutarate now has an amino group over here. Okay, so we now have our carbon skeleton. So these are what? Carbon skeleton. So we have a carbon skeleton, and then we now have what? The amino group transferred onto this molecule to form what? Glutamate. Okay, so let's say this is our glutamate over here. Glutamate. Okay. Now, this enzyme is what? Transaminase. So the transamination reaction is carried out by what? Transaminase. There are different transaminases depending on what? The amino acid involved. All right. Now remember that we are trying to transport what? Ammonia to where? The liver, because urea synthesis occurs where? In the liver, right? So the first reaction the cell has undergone is what? Transamination to form what? Glutamate. But glutamate is not a good transport form for what? Ammonia. So we have to transport it into a transport form for ammonia. What's that transport form called? Let's look at it, all right? Now, This glutamate, what happens to it is now transformed by the addition of what? An extra ammonia. So ammonia comes up and is packaged with glutamate. If you check, glutamate is a bicycle with what? Two seaters. All right, so there are two seaters. There's one seater and there's a what? Another seater. Now, we want to transfer another amino group to another amino group to this other seater over here, so that we would have what? Two amino groups. And then this would now be called what? Glutamine. And then the enzyme involved is what? Glutamine synthetase. Okay, so we have what? Glutamine synthetase. And then whenever I hear synthetase, what comes to your mind? ATP, right? So we use our ATP and then we have our ADP, right? And our inorganic phosphates. So we formed one transport form of what? Ammonia, which is what? Glutamine. And then the glutamine travels in the blood to enter where? The liver. So we have our glutamine over here. Now we have to look at the other transport form of what? Ammonia. This one is peculiar to what? The muscle cells. So in the muscle cell, this is a muscle cell over here. That's the skeletal muscle cell. We have, the muscle cell, skeletal muscle stores what? Glucose in the form of what? Glycogen. And this muscle cell is constantly what? Working to produce what? Pyruvate. So glycolysis always occurs in the muscle cells. And then, glycolysis forms what? Pyruvate. So we also have our glutamate over here. Remember, our glutamate is this boy over here with what? Two seaters, right? And then in one of the seaters for the glutamate, there is what? Ammonia or amino group in one of the seaters. Now, pyruvate is another boy over here. Let's illustrate that with the green, which has what? One seater. All right. Now, the one seater has not yet been occupied. And now, what this muscle cell does is that it takes the amino group from glutamate and transfers it onto what? Pyruvate. All right, because it has what? Abundant pyruvate, right? Now, we have a reaction occurring here, and this pyruvate would now take up what? The amino group from where? Glutamate. Pretty easy, right? And then the glutamate remains its two seaters without what? Any amino group. All right. So basically, this is the reaction that's occurred here, and then this now forms what? Our alpha ketoglutarate back. Right. So remember the transamination reaction, because this is also a transamination reaction where there's what? A pyruvate and there's what? Glutamate. So there is now transfer of amino group from glutamate to where? Pyruvate to form what? Alanine and what? Alpha ketoglutarate. Remember, glutamate could have been formed initially from this same reaction that occurred over here. So the same reaction would have transferred an amino group from amino acid onto what? Alpha ketoglutarate to form what? Glutamate, just the same way this occurred. And then what? Alpha keto acid, which now goes to what? Generate what? Energy over here. And then this glutamate, instead of going through the glutamine synthetase pathway, now combines with what? Pyruvate to form what? Alanine and what? Alpha ketoglutarate. And this reaction is also called what? A transamination reaction. All right. And the particular enzyme or the specific enzyme for this reaction is what? ALT. Okay. That's your what? Alanine transaminase. So we have our alanine transaminase or what? Alanine aminotransferase. Okay. Now we have our alanine being transported as well. All right. Now we have glutamine over here and we have what? Alanine over here. And then they'll now be transported into where? The liver for what? Urea synthesis. So we have our glutamine over here and then we have our alanine over here. Now these two molecules, are what? Transport forms of what? Ammonia. All right. We have a glutamine and alanine being two transport forms of what? Ammonia. Now, when these molecules get to the liver, what happens? Unloading. First we've discussed what the transport, and then now unloading of what? Ammonia. So we want to now, unload the ammonia from these pockets, right? Now let's discuss the glutamine first. Glutamine would undergo a reaction to form what? Glutamates. So we have our glutamate over here, right? And then this reaction is catalyzed by an enzyme found in the liver known as what? Glutaminase. Okay, so the name of this enzyme is glutaminase. So we have a glutaminase. And then this reaction is a hydrolytic reaction. So it uses water, like it pours in water to the reaction, right? And then to break it down to form what? Glutamate. And then what do you expect? Your ammonia, right? So one molecule of ammonia comes off. All right. Remember that our glutamate still has these two seaters, but then one seat is occupied. All right. So glutamine had two seaters, and the two seats were occupied. And then what? It gave off what? Ammonia to have what? One seat occupied in what? Glutamate. Okay. Now this glutamate is further broken down by an enzyme known as what? Glutamate dehydrogenase. Okay, so we have glutamate dehydrogenase. And then whenever you hear dehydrogenase, what do you expect? NAD. And then we have what? NADH. All right, to form what? Alpha ketoglutarate. Right, and then you expect another molecule of what? Ammonia to be puffed out, right? We form the ammonia, right? Now let's go over to the what? Alanine. Alanine can undergo a reverse reaction as that occurred over here. All right. So the reverse of this reaction occurs where? In the liver, where we have our alanine combining with what? Alpha ketoglutarate in a transamination reaction to form what? Pyruvate and what? Glutamate. All right. And then this glutamate can still be broken down by what? Glutamate dehydrogenase. Okay. Now, let me illustrate it over here. This glutamate can now be broken down by what? Glutamate dehydrogenase. Okay, to form what? Alpha ketoglutarate. And then we have what? Ammonia from alanine, right? So I just brought out this reaction over here, okay, for simplicity's sake. Now, remember that NAD was used and the NADH was given out, okay? But then this pyruvate over here can now, the liver is responsible for what? Gluconeogenesis. So this pyruvate can now be further used to form what? Glucose. Glucose. And this glucose can enter the blood, and through the blood can enter where? The muscle. All right. So I'm sure so many of you would have heard of what? Glucose alanine cycle. So this cycle is the what? Glucose alanine cycle, where alanine produced by what? Transamination reaction in the muscle will be transported to where? The liver. And then in the liver, the alanine was now used to form pyruvate and then to form what? Glucose, which is now transported back to the what? Muscle. Glucose alanine cycle involves what? Alanine being produced by the what? Muscle and then enters the way? Liver. And then in the liver, transamination reaction occurs to form what? Pyruvate and then to form what? Glucose. And the glucose is now transported through the blood into where? The muscle back. All right. So this cycle is known as this glucose alanine cycle, also known as what? Cahill cycle. Cahill cycle. All right. Great. So let's continue our urea synthesis. All right. So we formed what? Three molecules of what? Ammonia. All right. Now we have our ammonia diffusing into where? The mitochondria. So in the mitochondria, ammonia combines with what? CO2. All right, that's your carbon dioxide, and then the two of them together form what? A molecule known as what? Carbamoyl phosphate. All right, so we have what? Carbamoyl phosphate. All right, Great. So we have carbamoyl phosphate produced over here. And then the enzyme is what? Carbamoyl phosphate what? Synthetase. All right. So we have carbamoyl phosphate synthetase. You had synthesis. What do you expect? ATP, right? So it required what? 2 ATP, and definitely we have a 2 ADP and one inorganic phosphate because one phosphate entered here. And that's why he had the word phosphate. Okay. So we have a carbamoyl phosphate over here. This carbamoyl phosphate synthetase is an enzyme which is activated or regulated by what? A molecule known as what? N-acetylglutamate. So we have N-acetylglutamate being the what? Allosteric activator of what? Carbamoyl phosphate synthetase. Okay. Now we found that carbamoyl phosphate, and then this is the what? First step of the reaction. All right. Now, for subsequent steps, I'll tell you a mnemonic which we'd use to study each of the steps, right? The mnemonic we'd use is what? Old, colorful cats always ask for awesome umbrellas. So, old colorful cats always ask for awesome umbrellas. All right, we'll incorporate this mnemonic in the rest of the steps. Okay. Now for O, we have a baby over here called what? Ornithine. Ornithine is a baby molecule over here in the cytoplasm, right? So we have to transport it into where? The mitochondria, right? And then in the mitochondria, this transporter here is called what? Ornithine citrulline transporter, okay? So this ornithine is transported through this transporter. So the ornithine enters to react with what? Carbamoyl phosphate, all right? To form what? Citrulline, all right? So ornithine reacted with what? Carbamoyl phosphate to form what? Citrulline. What do you expect? Phosphate, right? So the phosphate leaves. We have our phosphate leaving. Yeah, right? So the ornithine combined with what? Carbamoyl phosphate, giving off what? A phosphate and then forming what? Citrulline. What do you think the enzyme would be? Ornithine carbamoyl transferase, okay? Or commonly known as what? Ornithine transcarbamylase. Okay, so the enzyme is what? Ornithine transcarbamylase. All right, so it transfers a carbamoyl onto what? Ornithine. Okay, cool. We're done with what? The second step of the reaction. Let's go over to the third step. This citrulline that was formed anyway comes out through this what? Transporter into where? The cytoplasm. So in the cytoplasm here we have our citrulline. Now, remember old colorful cards. So the old represented what? Ornithine. Colorful represented what? Carbamoyl phosphate. Cards represented what? Citrulline. Now we are going over to what? Always. This citrulline combines with what? Another molecule known as what? Aspartate. Aspartate. Whoa. Where is this aspartate coming from? Well, aspartate can be obtained from what? Our glutamate over here. So some of the glutamates would have combined with what? Oxaloacetate to form what? Aspartate and what? Alpha ketoglutarate. Okay, so some of your glutamates would have said, oh no, I want to form what? Aspartate, and then it would have reacted with what? Oxaloacetate to form what? Aspartate and what? Alpha ketoglutarate. Okay, so these are aspartate over here. Aspartate. All right, it's an amino acid. Now this aspartate combines with what? Citrulline to form a baby boy called what? Arginino. It's quite a mouthful, right? Argininosuccinate. Let's say AGS, right? And then the enzyme involved is what? Argininosuccinate what? Synthetase. Oh my God. We've heard what? Synthetase, right? And then when we hear synthetase, what comes to our mind? ATP, right? So ATP was used, and then we had our ADP. Now, and inorganic phosphates, definitely. Now, if you notice, 2-ATP was used in the first step of the reaction where carbamyl phosphate was formed, right? In the presence of what carbamyl phosphate was synthetase. And now, this is reaction 3 over here. We are using another what? ATP. So we are now using 1 ATP in reaction 3. All right. And also, if you notice, 1 ammonia was used in the beginning of the reaction to combine with what? CO2. And then I also told you that aspartate is an amino acid. What do you expect? It has what? An amino group. So it has ammonia, right? And then it donates 1 ammonia as well to the urea formation. Okay. So would eventually have our urea being something like this: CO and then NH2 NH2, right? So this one entered initially into the mitochondria to combine with what? CO2. And then this other one was from where? Aspartate. All right. And then this other one is from your CO2. Okay. Great. So let's continue. We formed our boy over here, which is the what? Argininosuccinate. Now, this argininosuccinate is now broken down, right? It's now broken down by an enzyme known as what? Argininosuccinate what? Lyase. All right, so to lyase, right? Now, the argininosuccinate is broken down into what? Tell me arginine, right? Arginine, and then it gives off what? A molecule which falls off the urea cycle, known as what? Fumarate. Whoa, fumarate. Remember, this fumarate can later enter what? The TCA cycle. This reaction is what? Reaction 4. All right? And then the last reaction, which is the reaction 5, is the what? Formation of ornithine back. And then urea is giving off. So we have our urea giving off, and ornithine is formed. Right? Urea is giving off. Then ornithine is replaced. Okay? And then this enzyme is what? Arginase. And it puts in what? Water. So there's hydrolysis of what? Arginine to form urea and what? Ornithine. So I'm sure so many of you will be wondering, okay, the memory aid, I'm lost. Don't be lost. Let me tell you. The old colorful cards always ask for what? Awesome umbrellas. Old for what? Ornithine. Colorful for what? Carbamyl phosphate. Remember, carbamyl phosphate would have been formed already from what? Combination of CO2 and what? Ammonia, right? Then cards for what? Citrulline, all right? So this is the citrulline which was transported out through the what? Ornithine citrulline what? Transporter, right? Now, always for what? Aspartate, ask for what? Argininosuccinate, for what? Fumarate, awesome for what? Arginine, and then umbrellas for our baby over here, urea, all right? Now, the mnemonic involved both the intermediates and then what? The molecules entering and leaving, all right? Now we found our big boy over here, the urea, right? The urea has to be what? Excreted. Now let's talk about what happens to the urea as it leaves the what? Liver. So we have our urea from the liver entering where? The blood. This urea needs to be what? Excreted. All right. This urea needs to be excreted, and then what happens? It enters where? The kidney for excretion into where? The urine. All right. And then some of the urea enters somewhere else Where do you think? It enters where? The intestine. All right. So some of the urea enters where? The intestine. So we have urea over here. And in the intestine, we have what? Intestinal bacteria. All right. So these are baby intestinal bacteria over here. And then this intestinal bacteria has an enzyme. This enzyme over here is known as what? Urease. Urease. When you hear urease, what do you think? It splits urea, right? So it breaks down the urea. As the urea passes through here, it's broken down into what? Ammonia and CO2, right? And then some of the ammonia will be excreted in feces, right? And then some of this ammonia would now be reabsorbed in where? The blood, all right? So we have the ammonia being reabsorbed in the blood. And then to now find this way to deliver for what? Urea synthesis, okay? And then the whole cycle occurs. All right. So now we've seen the fate of the urea. Is either excreted in the kidney or it enters the where? Intestine. All right. So when it enters the intestine, it's broken down by intestinal bacteria, which contains urease enzyme into what? Ammonia that can be reabsorbed into the blood. All right. And then some of the ammonia is excreted. Okay. Now something can go wrong here. Some persons can have what? Kidney failure. All right. So individuals with kidney failure can no longer what? Excrete urea. So what happens? Most of the urea is shunted into where? The intestine. So we have most of the urea shunted into the intestine. So we have what? Increased intestinal urea. And this bacteria now starts breaking down all the urea and then would eventually have what? Increased ammonia over here. And then would have what? Increased reabsorption of what? Ammonia. So we'd have what? Hyperammonemia. So hyperammonemia, increased ammonia in blood, right? So that's hyperammonemia, right? Now I'll tell you in a second why this hyperammonemia is very toxic to the body and is very bad. We need to deal with that, all right? So to deal with that, we need to give a drug known as what? Neomycin. So this drug is called what? Neomycin. And then what does this drug do? It's an antibiotic. So what do you expect? It destroys this bacteria over here. All right? Now, this drug destroys the bacteria such that whenever there is kidney failure and you give the neomycin, the urea that's been shunted to the intestine can no more be used to form what? Ammonia. And then hyperammonemia will not occur. All right? That's great. So let me tell you other forms of hyperammonemia before we now talk about the toxic effect of what this hyperammonemia. All right? The two forms of what hyperammonemia are what? Congenital and what? Acquired. All right. So when it's congenital, you know that there's what? A deficiency of the enzymes of what? Urea synthesis. Okay. So when there is deficiency of the enzyme, you know there are now what? Build up of what? Ammonia. Because the ammonia cannot enter the what? Urea cycle. All right. Because of the enzyme deficiency. Okay. And now the enzyme deficiencies are what? Autosomal recessive, except what? Are OTC, which is the most common enzyme that is deficient. That's the ornithine transcarbamylase. Okay, and this enzyme is the most common enzyme that is deficient, and also is what? X-linked. Okay, so it occurs more in where? Males. All right. Now for the acquired, maybe there could be a damage to the liver. Let's say liver damage, and you know in liver damage, what happens? Maybe there is what a cirrhotic liver. In cirrhotic liver, there is what? Portal hypertension, and there is now what? Shunting of what? Blood to where? Systemic circulation instead of what's going through the liver first. All right. So the ammonia can no longer enter the liver adequately, and then it now shunts into a systemic circulation. All right. And then maybe hepatitis. What happens? There's liver damage. That's the cells of the liver are what's damaged, and then they cannot undergo what? Urea synthesis. You can see other things like what? Kidney failure, which we talked about. We talked about the kidney failure where there is what? Damage to the kidney, and then the urea that has been formed cannot longer what? Be excreted. And then when it cannot be excreted, they now shunt into the intestine, and the intestinal bacteria break down the urea into what? Ammonia. Then there will now be what? Increased ammonia production. That's hyperammonemia. Right? That's why you have to give which drug? Neomycin, which now destroys the what? Intestinal bacteria. Right? Now, let me tell you what this hyperammonemia can do to your brain. In the brain, there are some cells called what? Astrocytes. Come on, I'm sure you know astrocytes. These astrocytes have what? Glutamate, right? And then they have an enzyme that can take up what? Ammonia from the blood. So we have what? Ammonia. You know, ammonia can cross the blood-brain barrier, right? Ammonia enters the astrocytes and combines with what? Glutamate in the presence of what? Glutamine synthetase. So we have our glutamine synthetase. Okay. So this glutamine synthetase can now produce what? Glutamine from what? Ammonia. Right? And this glutamine has a tendency to drag water. So it's what? Osmotically active, dragging water into where? The brain. Wow, what does this cause? Cerebral edema. All right. So we see the individual have what? Cerebral edema. Also, maybe from the eyes, there can be what? Blurring of vision. The mouth, for instance, there's what? Slurring of speech. And then there can be what? Somnolence. So you see the person sleeping a lot, all right? Somnolence. And then the person can eventually enter where? Coma, and the person can die. That's one major effect of hyperammonemia on the brain. And then when it's caused by liver damage, you can say what? The person has what? Hepatic encephalopathy. All right. So hepatic encephalopathy, maybe when there's a liver damage that now results in what? Hyperammonemia. And then these symptoms are seen. So it's hepatic encephalopathy. All right. Now, when we have all these, let's say we have our congenital hyperammonemia, or we have some acquired hyperammonemia, maybe from the liver damage and stuff like that. What do we have? In the blood, we have what? Buildup of something called what? Glutamine. Remember I told you that glutamine is the what? Transport form of what? Ammonia. Right? Remember I told you there's glutamine and there's what? Alanine. Right? Glutamine has the tendency to bind what? Two amino groups. Right? Now, this glutamine buildup will be high. The glutamine in the blood will be high, right? Indicating what? High ammonia, right? Now, we want to treat this hyperammonemia from either congenital or maybe liver damage. What happens? We give a drug. The name of this drug is what? Phenylbutyrate. This phenylbutyrate enters where? The blood, breaks down into what? Phenyl acetate. So this is a prodrug. It's, breaks down into the active form known as what? Phenyl acetate. This phenyl acetate then does something. It combines with what? Glutamine. So phenyl acetate combines with what? Glutamine, right? To form what? Phenyl acetyl glutamine. So phenyl acetate combined with glutamine to form what? Phenyl acetyl glutamine. This phenyl acetyl glutamine can now be what? Excreted easily. Okay, so you can see that we are now excreting phenyl acetyl glutamine, thereby excreting what? Two molecules of what? Ammonia. All right, so that's a pretty good treatment. All right. So guys, we've come to the end of this lecture. It was a long ride. I'm sure you enjoyed it. Do well to subscribe, put your comments below, like, and then share to your friends so they can benefit from this lecture. Thank you, and see you in the next lecture.